universal recipient, the one that can take blood from any blood group, is the blood group AB. Why? Because it does not have any antibodies present. The O blood group cannot take. Don't you remember? Yeah, yeah. I discussed, like, I even asked the blood groups of
Yeah,
the students.
yes, ma'am. I remember.
Remember?
I remember
Hmm.
that.
All right. Now, so this is how the crossing of the two blood groups can be done. And remember that the parent, if a parent is AB blood group, or the this kind of, like, the genotype may differ from all the organisms. So as to check on that, you have to make the crosses, right? And how do we make crosses? We cross this with this, and then this with this, right? And then same way the whole crossing will go around. For example, IA and IA. So we have one genotype. Then IA and small I. So we have the second genotype. Then IB and IA. So we have this one. And then IB and small I. So we have four genotypes. So this way only you will make the crosses. Now here, since A and B blood group, both of them are present together, or both of them are expressed together, that is why we call this type of inheritance as the codominance. And it is one exception to the Mendelian law of inheritance. Now, moving forward, we'll talk about the inheritance of two genes, just like how we discussed the inheritance of one single gene, and that was the monohybrid cross. Now, we'll discuss about the inheritance of two genes, and that we call as the dihybrid cross. Now, when the two characters are taken into consideration and then the crossing is done, we call it as the dihybrid cross. Now, for this, a cross was made between a pure, meaning a homozygous brown yellow seeded plant with a wrinkled green seeded plant. Now, you see, round and yellow. Round was the shape of the seed, yellow was the color of the seed. So, in this crossing, we are taking two characters into consideration. And that is why we are calling it as a dihybrid cross. Okay. Same way, we are taking wrinkle, which was the shape of the seed, and now we're taking green color, which is the color of the seed. And then we are crossing them together, and that is how the dihybrid cross is done. Now, capital R, capital Y. Capital R represents the round shape, and capital Y represents the yellow color. Both of them are the dominant ones, right? And the small ones represent the wrinkled and green seed. Now, when we cross them out, the gametes from the parent A is capital R and capital Y, and the gametes from the parent B is small r and small y. Now, when these fuse, then F1 generation is formed, and F1 generation is the, what do we get? Capital R, small r, and then capital Y, small y. Now, what do? We do selfing to find out the F2 generation. Selfing means self-fertilization. Now, when we do so, then we get, like first the selfing process, since both of the genotypes are same, we take out the gametes yet again. Now, the gametes here will be capital R, capital Y, the first gamete, then capital R, small y, meaning if capital R, capital Y, and small y are there, then this is the first gamete, right? Then this is the second gamete. And then small r, capital Y, then small r, small y. So this is how we take out the gametes. And then we make a Punnett square with them, wherein we write one set of gametes on the top and one set of the gametes on the side, and then we do the crossing. So when we do capital R and capital Y with capital R and capital Y, we have R, R, Y, Y. Then we do capital R, capital Y with small r, capital Y. So we have capital R, small r, capital Y, capital Y. Always remember that the dominant trait will be written first, no matter what is happening. For example, when we are crossing out capital R, capital Y with small r, capital Y. So we will write like this. Always remember the dominant one will go first. So this crossing thing, you people are understanding how we are finding out the cross, the F1 generation and the F2 generation. Now, when you like I have left this so that you people will do it when you write it down. But when we finally do this, we find the phenotypic ratio of the dihybrid cross, and it comes out to be 9:3:3:1. This is the most important thing. Like up until now, what was the ratio we were receiving? We were receiving 3:1 ratio, but now we receive the 9:3:3:1 ratio. So this was about dihybrid cross. Now talking about another law, that is the law of independent assortment. Now, in
Ma'am,
this, yes,
I have a doubt. In the
hmm,
exam, will they ask the phenotypic ratio or the genotypic ratio?
for dihybrid cross, always the phenotypic ratio is asked, because if you look at these offsprings, there are like so many. It'll be very hard to find out the genotypic ratio. So mostly phenotypic ratio is asked, meaning the
And for
external
monohyte?
observable features. Monohybrid, both of them can be asked because it is a smaller cross. That is why.
Okay, ma'am. Thank you.
Now, the law of independent assortment, it says that if two pairs of contrasting characters, meaning four traits altogether are present, each pair will segregate or will get assorted independently from each other. Why? Because now we are seeing when we are making the crosses, the round is also shown in the offspring and so is the yellow. So in one pair of the trait, both the characters, they are being you can say inherited separately. They're not getting lost, they're not getting masked, so they get inherited or they segregate independently from each other. The round is also expressed in the offspring, and so is the yellow. This thing is clear up till now? Clear to everyone?
Yes, ma'am.
All right.
Yes, ma'am.
Okay. Now talking about another factor, that is multiple allelism. When there is the presence of more than two alleles for one gene, then that we call as multiple allelism. For example, normally for our gene, that is height, right? For height, we have only two alleles. We have a tall allele and then we have a dwarf allele. But when there will be another and more than that, so that is multiple allelism. Okay. For example, like there is a case of uh, start synthesis. So capital A, and also for this thing, humans also. Capital A, capital B, and capital C are used for the inheritance process. So more than two alleles, if they are present, then that we call as multiple allelism. Clear? Okay. Now, if you look at these many characteristics, if these characteristics that I mentioned here, dominance, codominance, multiple allelism, incomplete dominance, and polygenic inheritance. Out of this, which all are found for, you can say, ABO blood grouping? Tell me. And these are the options. You have to choose one from the options. Like how many of these phenomena are found in the blood grouping system of you?
Ma'am, should we answer in the chat or should we say it verbally?
You can tell me the option that you're choosing. Which option you're choosing?
Option 2, ma'am.
Option 2. A, B,
Yes.
and C. Correct. Dominance, how? Because one trait is dominant over the other. Codominance, because A and B both are present together. And multiple allelism, because A, B, O blood grouping, more than two alleles are there. Very good. What about the others? Did you people understand this question? Hmm?
Yes, ma'am.
Okay. So, are like all of you are sleeping? Eh? Where are you all? Why? Like I have asked 3 times to switch on your video. Why no one has except for one student? Hmm? Only one student has switched on his video. Rest all are not. What? You don't want to switch on today? You don't want to show me your faces today? Also, did you people have your test?
Yes, ma'am.
How was the test?
No, ma'am.
Fine.
You did not attempt? Yeah, yeah.
No, ma'am. Not attempting today.
Okay. Yes, Khadija, your test was good?
Yes, but I didn't really write like a lot of details. And this chapter is kind of confusing.
Which one? The genetics?
This one. Yeah, I don't like it.
Yeah, yeah. You have to learn a lot in this particular
Yeah.
chapter. But don't worry
Ma'am,
about it. Hmm.
ma'am, comparatively, this is easier.
You find it easier? It's
Yes,
from
ma'am.
student to students. The opinion can vary from like personally, I did not like this chapter too. But see, one more thing, like right now we are doing the chapter for the first time, right? So you will find it difficult. Now let's just do one thing, that once this chapter is over completely, we'll do a
Yeah.
revision of this also. And in that
Okay.
revision, it doesn't mean that only I will speak. I will ask you and I will teach you at the same time. So you people have to learn side by side. Okay.
Okay.
Yes, and in the class also, if you did not understand anything, you can ask me, I can explain again. All right. Now, this was the dihybrid cross and everything is completed. Now we come on to the next topic, that is the chromosomal theory of inheritance. Now, everyone knows what chromosomes are? Can someone explain what chromosome is? Explain what is chromosome. When I ask, like, do you people know? You like, yes, yes. And when I say explain, then everyone goes blank. Tell me what chromosome is. Now, this is from 11th. This is not even 12th. So, tell me what chromosomes are. Tell me their shape.
They're like thread-like structures, ma'am.
Thread-like.
Yes.
Okay. Okay, what else about them? See, chromosomes are mostly rod-shaped structures, not thread-shaped. Okay. Yes. You will tell me, I will not go forward. Students, what is a chromosome? This so you should know, because you people have passed class 11th and now you are in class 12th. Tell me what is chromosome. Khadija, what is chromosome? Also, still, like, so many of you have not switched on your videos. Why? Just because
It
I'm
is an
friendly in the class,
organic,
you will not listen to me.
ma'am. Is it an organic genetic material?
No,
No.
no, big no. Genetic material is DNA or RNA. But you are very close. It's close to genetic material. No one can tell me what chromosome is. Amina, what about you? Hmm. Siddhika, what about you? Yes, Amina.
A nucleus, sorry, inside a nucleus.
Say it clearly. I did not hear.
A structure formed inside a nucleus.
Correct. Okay, good. We have found that chromosomes are present inside the nucleus. Okay, very good. At least something came. Now, what do they do inside nucleus, students? Anyone? Umar, what about you?
Make protein and DNA.
Hmm?
Make protein and DNA.
It makes protein and DNA. No, it is
No.
made up of protein and DNA. It does
Oh,
not
sorry.
make it, it's made up of it. And see, oh God, this is very bad, students. This is so bad. Listen, chromosomes are the carriers of DNA. It's even written right there on your screen. You could have even cheated and tell me what's written on your screen if you had used that much of brain. Chromosomes are the rod-shaped structures that are present in the nucleus. They are the ones that carry the DNA molecules. They are the ones that are the physical carriers of DNA. Okay? Clear to everyone?
Yes, ma'am.
Yes,
Okay.
ma'am.
Good to know that. Okay, still many students' videos have not been on. Why? Why? May I ask why? Is there any particular reason? So you can tell me the reasons and I'll maybe give you the benefit of the doubt. Or else there's no doubt, no benefit. Tell me. Hmm? No response also. Why? Why students low on energy today? You guys are low on energy? You don't want to study genetics? You just want to study animals and Arey Baba, speak. Say something. What has happened? You are ignoring me today. Huh? All of you are ignoring me.
No,
Why
ma'am.
so? You're like, let her speak. She always speaks. Eh? Tell me what is happening. Do you like? Are you not liking the chapter? You liking the chapter? Then what's the problem? Maybe
Ma'am,
the
today's a
problem is me.
Saturday. No, ma'am. I think today's a baby Saturday. That's why no one is in the mood of studying.
Is it? Everyone in the mood of like weekend partying, huh? Party at home, not
Saturday is
studying
a
in it.
very lazy day.
Saturdays are very lazy days.
Yeah,
Yes, ma'am.
we have
Very
school
late.
the next day.
Okay, but what to do? Your classes are scheduled on weekend only. Like
Yeah.
we can't do anything about it. So I need energy. Now from the next time, please have something, energizing, some caffeine or something and come and sit in the class then. Maybe you people will have more energy like that. Umar, are you
Okay.
there in the class?
Yes, ma'am.
He's there. Okay, it's good to know that you people are there, even though you are doing your best to ignore me and not switching on your videos. I'll not continue the class if everyone's video is not turned on. I'll not continue the class. You are my senior most batch students. I don't want to scold you. So please spare me that. I am the cool teacher. Let me be the cool teacher. I don't want to be the traditional strict teacher. So don't make me one. Switch on your videos, everyone. That's like it. Good, Ikra. Now, like, do I have to treat you like kids? Like, oh, very good. Star to Khadija, star to Ikra for switching on the videos. Toffee. Umar and Siddika, you are the only ones that are left. Come on. Be good students. Don't make me the bad teacher. Okay, very good. Siddika, very good. Okay, Umar. See, switch on your videos because every once in a while I should know what you people look like. Or else I'll never know. Like, maybe you people do so well in the exams and your, images are, printed in the posters and everywhere. And I'll like, oh, some student has done very well. Now someone else will tell me, ma'am, this is your student only. Please don't make that happen with me. I should know what my students look like, isn't it? Maybe one day you people will make history and I should be able to recognize you so that I can say, oh, see, that's my student. All right. So have I, you can say, entertained you enough that you people will stay interested in the class? Okay. From time to time, if you need entertainment, tell me, okay? Just pay attention in the class and tell me if you need entertainment. I'll entertain you. Okay? All right. Now talking about the chromosomal theory of inheritance. Now I told you what chromosome is, right? Now these chromosomal theory of inheritance, this states that whilst the DNA is being transferred from one generation to another generation, it is not being transferred alone. Chromosomes are actually playing a great deal there. Now, Mendel started his work on the pea plant in the year 1865, but his work was not recognized until 1900. Meaning, up till like from the point that his work became popular, it was not recognized. People were not giving value to him. Poor Mendel. Why? Because first of all, in that point of time, communication was not that good. I mean, there was no Instagram and there was no social media, nothing at that point of time. So his work, whatever he was doing, it was very hard to reach a greater audience. Okay. So, and apart from that, since his work was not reaching the greater audience, it was not possible for many people to know about his work. Secondly, we know that he was the first person to actually use mathematics in biology, mathematics and the statistical knowledge in the biology. Now, for us biology people, we do not like maths as it is, right? We do not like maths. That is why we choose biology, because almost all of us suffer from maths a lot. I mean, for me, I can speak, I suffer a lot. I hate maths. I cannot hate anything more than maths. But so same goes for all the biologists, right? So and in his time, since he was the first crazy person to use maths in biology, so no one was accepting it. They were like, how could you use maths in biology? It's not acceptable. So that is why his work was unrecognized. And apart from that, even though he gave this whole entire theory, now the theory, any theory, if you give, it's useless if you do not provide a proof for it, if you do not provide an experimental proof for it. So he did say that, okay, factors were being transferred and everything, but he did not say anything about how and what the factors were. So there was no proof. So again, because of that, and at that point of time, chromosome, the mitosis, the meiosis, meaning the cell division and chromosome, this was not discovered in his time. So he was, you can say, way ahead of these discoveries. So these are the four reasons because of which Mendel's work was unnoticed. This is a very like great question. Like even though he did such a great work, why was he unnoticed, right? Now, in the year 1900, there were three scientists, De Vries, Correns, and von Schermach. They independently rediscovered Mendel's work or Mendel's result on the inheritance of characters. Now, they were working alone on whatever topics that they chose, and they independently, three of them separately, actually found out the results that were found by Mendel. Also by 1902, the chromosomes were also discovered. Now, since advancement in the terms of, you can say in the field of research was done, so chromosome was discovered. Now, Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes. And they used chromosome movement to explain the Mendel's law. So according to Sutton and Boveri, they said that the pairing and separation of a pair of chromosome would lead to the segregation of a pair of factor they carried. So if like two chromatids make up the chromosome, so when the chromosome separated, the DNA, the genes that they were carrying, they also separated. So that is what they said. Now, the knowledge of the chromosomal segregation, which happens during the cell division, and the principles of Mendel, when you combine them together, sort of like a collaboration, then that becomes the chromosomal theory of inheritance. Clear? In the chromosomal theory of inheritance, we are seeing as to how the chromosomes are playing their part in terms of inheritance purpose. Now look, this part up till now, is everything clear to everyone?
Yes,
This whole
ma'am.
entire story? Okay. Now talking about the features of the chromosomal theory of inheritance. Like the hereditary traits, meaning the genes, the DNA, the chromosomes also retain their number, their structure, and their individuality. Meaning, once the chromosomes are passed on from one generation to another generation, it doesn't mean that the chromosome will change. Just like how the DNA does not change, same way the chromosome also does not get changed from generation to generation. They neither get lost nor do they get mixed up. So this is the property that is shared by the chromosomes and the genes similarly. Now, now these are the points of chromosomal theory of inheritance. Both chromosomes as well as genes occur in pairs in the somatic cells, meaning in the body cells that are diploid. So these chromosomes and the genes, they are present in pairs. Now, the two alleles of a gene pair are located on the homologous sites on the homologous chromosome, meaning they are located on the same chromosome. Both the chromosomes and the genes, they segregate at the time of gamete formation, such that only each pair is transmitted to the gamete. Why? Because we know that during gamete formation, meiosis takes place. And when meiosis takes place, there is a reduction in the number of chromosomes. Or you can say from diploid, the cell becomes haploid, right? So that is why the chromosome number also gets half. So does the DNA content. Any doubts up until now? The points of chromosomal theory are clear to everyone?
Yes, ma'am.
Surely clear, or any doubts are there? Clear to everyone? Khadija, clear? All right. Now, this was the theory of chromosomal inheritance, right? But now, again, as I told you, no theory is valid without proof. So the experimental proof or the experimental verification of the chromosomal theory of inheritance, it was given by T.H. Morgan, Thomas Hunt Morgan. And he worked with Drosophila melanogaster, that is the scientific name of the fruit flies. So he worked with the fruit flies and he wanted to explain the theory of inheritance. Now, why did he choose Drosophila? Because Drosophila can be grown on simple synthetic medium in the lab. They have short lifespan, meaning they only survive for 2 weeks. A single mating produces hundreds of offsprings, and the male and females are distinguishable. And they have smaller number of chromosomes. They just have 4 pairs of chromosomes. Clear? So they have in totality 8 chromosomes only. So this is like, these are the reasons why, just like how there were reasons why Mendel chose the pea plant, same way there are reasons why Thomas Hunt Morgan will like he chose the Drosophila. Any doubts up until now? Clear to everyone?
Yes, ma'am.
Yes, ma'am.
Okay. Now, students, I did say that I will do a revision once the chapter is done, but that does not mean you'll rely on revision. Okay, pay attention here also, and in the revision, it will be a proper revision. Okay, now you people can note it down. Okay, this chart or this phenotypic, genotypic thing was noted down by you?
No,
The
ma'am.
blood group?
We did till
No,
test
ma'am.
cross, ma'am. We did till test cross.
You have done only till test cross.
Yes, ma'am.
Yes, ma'am.
We didn't write incomplete dominance. We didn't write co-dominance anything.
Okay, now you people can note down. You can switch off your videos, write down comfortably. When I ask you to switch off your videos, you people do it very quickly. Huh? But when I ask you to switch on your videos, you're like, will not do it. Anyway, you can switch off your videos and write down comfortably.
Done, ma'am.
Everyone's done?
Yes.
Done, students?
Done, ma'am.
Everyone's done?
Yes.
Done, ma'am.
Everyone's done? Hmm?
Yes, ma'am.
What about the others? Are you people writing?
Yes, ma'am. Done, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Done, ma'am.
Also, everyone complete this crossing square, okay? You have to write all the genotypes on your own. The ones that I've left, you write it, okay?
Okay, ma'am.
Okay, ma'am. Done, ma'am. Done, ma'am.
Is everyone done?
Yes, ma'am.
Ma'am, should we write this question? ABO blood groups question.
Yes, yes. It's a very famous question. You know, this question came in NEET. So everyone, you can write that. Like, which of the following does the ABO blood group shows?
Done, ma'am.
Everyone's done?
Yes, ma'am.
What about the others?
Yes, ma'am. Done.
Done, ma'am.
Is everyone done?
Yes.
Ma'am, what's the name of Walter Sutton?
Sutton.
Sutton.
Sutton. Sutton.
And Theodore Bovary.
Bovary.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Okay, students, you note down till here only for today. We'll keep it till here only and we'll continue the rest in the next class. Everyone's done?
1 minute, ma'am. I'm writing the first one.
Okay.
Ma'am, just a minute.